So I was today on the DEM website. Do you know DEM?
DEM?
Yeah.
What's that?
That's the designated emphasis on biotechnology.
Oh,
and that's not your name.
Oh, DEB.
Oh, DEB, yeah.
Yeah. Oh, okay, yeah.
Are you there?
Yeah, I'm doing the biotech one too.
Okay.
Yeah, I'm doing the biophotonics one and the biotech one. They're separate.
Oh, they're different?
Yeah.
They're two different things?
Yeah, there's the DE in biophotonics, and there's the DE in biotechnology.
Okay.
The biotechnology one is much more extensive. You have to do a 3-month internship, which is pretty cool because they help you set it up. So it's a little more of a commitment than this one. But have you taken your qualifying yet?
No.
Oh,
that's my first year, so...
okay, yeah. Well, you're in a good position to do that now. See, I joined those late, so I had to push my qualifying exam back by a whole year.
Oh, really?
Yeah.
Because you had like one requirement.
Yeah, because the main class that I'm taking right now, this MCB 263, you have to have, and it's only offered once a year, winter quarter. So if you don't do it like right away, then you kind of have to, you know.
But what do you think are the advantages to have that?
Well, it depends on what you want from it. I was just trying to get more experience in that area. It's a lot of genetic engineering is kind of the main focus of the biotech thing, and I want to learn more about that because my graduate group doesn't really focus on that. So, then plus the internship, you know, if you want to experience a little bit of industry, what it's like, you do that internship. So, I've heard they've said that some people that have come out of grad school with the DE in biotechnology, they didn't have to do a postdoc. They actually got a job in industry based off their internship that they did. So, if you think you want to go into industry, it's a good thing.
Somebody else told me if you want to go into industry, that would be definitely a good thing.
Yeah. It should be on your transcript, you know, your degree plus that. That's pretty cool.
Yeah. Did you also apply for the fellowships there?
I haven't done that yet. I didn't qualify until this year because you don't qualify unless you've taken, I think, that main class and been enrolled in the program for a certain amount of time. Last year I didn't qualify for it, so I'll probably apply this year. But I don't know how competitive they are. I have no idea. So, but to do the DEB though, anybody can join that. Yeah. So definitely a cool thing. I mean, well,
what happens to me?
Yeah, there's a year-long seminar you have to do, which is like an hour every Friday.
But what do you do there exactly?
Not much, just
listening to
talks and then write up a little one-page, half-page thing on the talks. That's not hard.
Same as your
main time-consuming one is the main class, Fundamentals of Biotechnology, I'm taking right now. The qualifying exam, you've got to have somebody from the biotech that's a member of that biotech BE on your qualifying exam committee. So you're going to be quizzed on it. But that won't be too
qualifying exam.
Yeah.
And he's going to...
Yeah, so, but most faculty are...
But some from the committee, from the department, but they're like all from different departments.
Yeah, they are. So the DE in biotech is not a department, so it's like Thomas Huser is going to be on your boss is going to be on my qualifying exam, and he's going to do the DE in biophotonics quizzing for me to get the DE in biophotonics. And Charles Bevins, one of my other mentors, he's a member of a lot of faculty are members of the
everybody, almost, I checked it today, because I just got today.
Yeah, a lot of people are part of the biotech.
Like almost like everybody from my department is on the board actually. So.
Yeah, so it should be a problem. And then just whether or not you want to do that 3-month internship, you have to do that. So that's 3 months of time away from your research you have to take. You know?
But I don't think that's a problem. No. That's actually a good thing.
For me, it's not, but some people think that is.
But you can also choose where you do it?
I think so, yeah. It sounds like it.
You can also do it abroad.
You know who I would talk to is Dr. Judy Chelstrup. She's the coordinator for the DEB. She's a really wonderful lady. Just go and tell her you're interested and ask her about it. Or tell you a little bit about it. I'm not sure it's something you want to do. But yeah, it's a pretty cool thing. I think it's worth it. But I think you have to decide. You know, some people want to get out of here in 4 years. You know, they're like, I don't want anything to slow me down. You know, it's definitely going to slow you down a little bit because you got to take more classes. You know, you've got to. Well, I'm slowing down even more because I'm doing this one too. And they don't cross correlate. They're completely separate. The DE and biopharmaceutics. the DE and biotechnology.
But this DE in biophotonics, that's going to be...
Is what now?
How is this working out?
It's much different. It's another year-long set of seminars.
What type of seminars?
Do you go to the biophysics seminars on Friday?
No.
It's like $290, I think. $290. Yeah, it's just 1 hour every Friday, and that's just attendance. I think is all you have to do for that. So I'm guessing because you're in the biophysics group, right?
No, biomedical engineering.
Oh, okay. A lot of the biophysics graduate group students will do the DE in biophotonics because a lot of those courses correlate. Like the biophysics seminar, it's the same. You know what I mean? Yeah.
But so for the biophotonics, you need this course here?
They give you a list of courses.
How many are there?
There's a pretty big list. You have to have one in the sort of the biological sciences and one in the biophotonic sciences. So I took membrane biology, which was like biophysics 241 as my biology one, and I'm taking this as my biophotonics one. Okay. So again, it's 2 more courses plus a year-long seminar, plus getting quiz on your qualifying exam. If you have to have Thomas or somebody. I don't know if it's a rule against having your PI and might not be able to have Thomas and might have to have Frank or somebody.
Yeah.
I don't think that's a big deal.
And then it's just going to be written on your.
Yeah, I think it's on your degree and your transcript. I don't know if it's on your degree too.
I thought it's on the degree. They said it's going to be
your degree,
PhD, blah blah blah, and then there's extra.
Definitely, it's a good opportunity. If you're not trying to get out of here in 4 years and you don't mind slowing down a little bit, I think it's good. Some people they don't want to get out of here.
But you don't have to take do an internship if you do.
No, there's no internship with the biophysics. I kind of want to do my internship with the biophysics because I want to learn the techniques. But you can do it like Genentech. You can do, there's lots of different.
That's interesting.
You can kind of guide it towards what you want.
The biotech EE?
Yeah, tell them about the internship.
Yeah, I just heard today about the other department, the designated emphasis on biotechnology.
There's
and so I was interested.
Yeah, that one definitely had a clear goal.
Yeah.
That EE. When we set up the biophotonics DE, it wasn't quite sure what are the goals of doing it. Basically, Dennis Matthews said, going on the record. Basically, he just wanted to set up as close to an academic department as he could in biophotonics. No matter what the content was, he just wanted to have something called the Department of Biophotonics. The closest we could get right now is a designated emphasis in biophotonics. So it's good. Just get it done. I don't care what you do or how you propose to do it, just do it.
Okay.
So they set one up, but it wasn't necessarily set up with any specific mission or goals in mind. And so as a result, we're still kind of thinking about, what do we want to accomplish with the DE. So we offer some courses, but it's not as well thought out as the biotechnology designated emphasis. So there, they had a clear vision that this was an opportunity for graduate students to get exposure. Ultimately, it's an internship program. The whole DE seems to revolve around ultimately training you and preparing you for doing an internship in industry, right? Yeah. So now that we've seen how it works over there, it makes sense for us to try and do the same, and I think we will. Whether or not it's done just within about photonics DE, or whether we just do it as a CBST activity. Maybe it'll be a CBST activity.
Okay.
And lately too, I've been getting all these emails from students from China, from France, interested in doing like a summer research experience. These are sometimes undergraduate, also graduate students. And so we're trying to figure out how to maybe set up an international exchange program.
Someone told me actually about maybe an exchange program between France and
Yeah. So
I thought that also students from here could go
to France.
Yeah.
Maybe. But how's it going to work for you? I don't know.
I would go.
You would go.
Sure. Over the summer.
Okay, so today we're going a little off schedule. What happened was that Thomas Huser was originally scheduled to give a talk about nonlinear microscopy.
He's slacking.
What's that?
He's slacking.
He's slacking. No, he's not slacking. He's completely overwhelmed. He's a problem. We have this week a special workshop on the picoquant single photon counting system. We do it every year in January, and it turns out that the workshop starts up again this Thursday and Friday. It's scheduled to precede the SPIE International Optics, what's it called? International Society for Optical Engineering, right? And this is Photonics West, which is down in San Jose. And it's a big event. Usually everybody comes to attend that, and so we figured while everybody is in California and nearby, we would hold the picoquant workshop. So that's the reason for the timing, and Thomas is in charge of that workshop. And so between that and also he's coaching 2 of our students at CBSD to give their presentations at the SPIE conference next week. Next week. So he just felt like he couldn't do a good job, or he couldn't focus if he were to give a presentation today. But he's going to take the spot one of the 2 spots that I had left open. So I think Thursday, February 13th, or February 12th is going to be the date that he's going to take over instead. So what I'll do today, once I get this computer hooked up, is to give you, I was looking over the list of topics and discovered that we really don't have a whole lot of material on optical labels. And that's another big section. I mean, you've heard now about fluorescence imaging, but we haven't really talked much about fluorescence labeling and what are fluorescent labels. I don't know if you guys, how much experience have you had with fluorescence staining? Have you done any?
No, physical, hands-on, no.
Okay.
I've studied it.
But you haven't done it. And so, like working with antibodies, do you have that background, or do you know about it?
I did antibody stuff in cell biology.
Maybe a little bit.
All right. So maybe this will be a review, or maybe you'll get some material. But my own background is in immunobiology, and so kind of got into this in depth. I did a lot of cell imaging and fluorescence staining. So that's why I thought I'd give you this overview. There will be a couple of more advanced talks. For example, the engineered phytochromes, which are plant proteins that have been engineered to become fluorescent, right? Which could be an advanced molecular probe. But that's going to be a separate talk, and it doesn't really cover the basics about what is a molecular probe and how do you create a fluorescent label. So that's going to be what we'll be about. Okay. Since we didn't decide on this plan until last night, I just finished working on this presentation 5 minutes ago. So I apologize if it's rough. Also, I uploaded the presentations on the slide to the
Yeah, I got the file shape.
So the link that you had before still works. Is that right?
To the shared
Because
folder.
I didn't put up a new link to.
I didn't check.
Okay.
It seems to work. The shared file folder.
Yeah. Okay. So I wasn't sure if that link would expire. I told it not to let it expire.
Yeah.
But I'm never too sure what that thing.
Yeah.
Okay, so, is that okay? All right, so this ought to be short. Hopefully we'll get to that a little earlier than 3 o'clock today. This talk is an introduction to optical labels and molecular probes. And the goals basically are 3. First of all, to introduce the field of biodetection. So by way of introducing you to molecular probes, I'm giving you an application first, and this is the field of biodetection. Okay. And then this will be the rationale for the need for developing advanced optical molecular labels. And then to describe some of the essential features of immunofluorescence, which includes understanding what antibodies are about, what's their structure and function. And then we talked about fluorescence already, so I won't delve too much into that. But just to introduce you to the idea that immunofluorescence brings 2 technologies together. One is the optical side, which is fluorescence, and the other one is the immunology side, which is the antibodies. And by coupling those 2 features, you create the special molecular tag that's extremely sensitive. The classical application for immunofluorescence is called the ELISA fluorescence assay, and the Sandwich assay. And so Steve Land has mentioned this briefly, but I'll go into it in more detail. And then finally to discuss some new and alternative strategies for molecular detection. And that part I didn't get to completely write out, so I'm going to kind of ad-lib out towards the end. Right? So first of all, introduce you to one application area, which is my area of expertise, which is infectious disease. Right? Basically, in this domain, infectious disease has always plagued mankind. These are actual pictures of people who were infected with smallpox. Right? And they believe that smallpox is something that emerged about 10,000 years ago, so almost as long as mankind has walked on Earth, there has always been some kind of infectious disease in existence. So the proof of that was looking at the mummies that are in some of the Egyptian museums, and by looking at seeing the appearance of scars that very much resemble smallpox, they believe that smallpox existed for that long. And so smallpox is caused by something called the variola virus. The reason for some of these topics was I was working at Lawrence Livermore's lab before I came here to UC Davis. And for us, infectious disease was not only a medical problem, but a security and defense problem. So there's a history of biowarfare talking about how back in medieval times, people knew that infectious disease could be used as a weapon. And so they would actually take the bodies of people who died from infectious disease and throw them over with a catapult into, say, a castle or a fortress, using basically dead bodies as biological warfare to infect the people inside the castle. Even in periods back in the Civil War, there was use of different gases to attack other people. And then even in Japan, there was this group, a terrorist group, who wanted to try and use anthrax as a way of spreading anthrax in the Japanese subway system. Unfortunately, they weren't very well informed, and what they ended up doing was vaccinating everybody against anthrax, because they were using antibodies. They didn't understand what they were using, so they were using anti-sera against anthrax, and not the anthrax bacteria itself. And so they had the right idea, but they had the wrong ingredient to try and cause mass destruction. And so these are all just images relating to bioterrorist activities in the past. So here I was just explaining to people what Livermore's biodefense effort was about. Don't need to worry about that so much, but the goal of biodefense was to protect against bioterrorism, which is the deliberate use of microorganisms or toxins to reduce death or disease in humans. And the main purpose of biodefense is to prevent the development and the use of biological weapons, as well as to help relieve the suffering that would result if prevention was failed. And so with infectious disease medicine, there were two tools that really helped us to combat it. And one is vaccines, and the other one are antibodies. And are you guys clear on? I'm sorry. Oh, this is a mistake. This should be an antibiotic. Right. So, if I were to ask you what's the difference between a vaccine and an antibiotic, could you explain?
Antibiotic kills the microbe directly, but a vaccine will use your own immune system to fight it. Right.
And so, in general, vaccines work against viruses. Antibiotics would not work against viruses. We credit two scientists for this.
Frank, why is it they can't develop an because literally the definition of antibiotic, right? I mean,
they're antiviral. They're antiviral medicine, right? But that hasn't been very tremendously successful. It's all very specific. They haven't found common mechanisms that are effective against a broad spectrum of viruses. So you can develop a drug against one type of virus, but it won't work on another type of virus. Whereas bacteria seem to have more common physiological mechanisms, and so because of that, you can develop drugs against a whole class of bacterial organisms. Viruses, it's partly because they're so simple, right? It's just a encapsulated piece of genetic material, and so what the disease that each virus causes is totally different depending on what the genetic material is inside. Otherwise, what else can you attack that's generally applicable is not so clear. So that's kind of one reason why antiviral medications are hard to drug. There's two scientists that we credit for the vaccines and antibiotics. One of them is Edward Jenner, who was a British physician who had made the observation back when smallpox was still a major problem that women who were milkmaids had developed a similar disease called cowpox. All right, and so cowpox is one where women who are milking cows develop these bumps and sores on their hands that look like smallpox. But he noticed that women who had cowpox never seemed to contract smallpox. So something about cowpox prevented them from getting smallpox, and he was interested in, he hypothesized basically that there's something in cowpox that keeps you from getting smallpox. So he did this one classic experiment that would never be condoned in today's environment and society. He took basically the watery kind of the pus that came out of that seeped out of the bumps of a milkmaid's hand, right? And he used a needle to deliberately poke this into a young child who had never been exposed to either cowpox or smallpox before. Okay. So this is kind of a painting showing him doing that, all right? And then, sure enough, the kid developed symptoms of cowpox. Okay, and so his hands developed the same rash bumps that the milkmaid did. But then when those symptoms subsided, he took that kid and thrust it in front of every single smallpox patient he could find. All right, and the kid would not develop smallpox. So he called this process vaccination because the Latin word for cow is vacca. And so to vaccine was basically to introduce something of a cow, to kind of use the powers of the cow to protect the person against smallpox. Okay, and so that's what that process was about. So he developed the first vaccine, or was first documented. People in China say that they knew this idea a long time ago, but there's no proof of that. So we credit Jenner for this. Louis Pasteur was a French chemist who discovered the germ theory of disease. So people knew the concept of infectious disease, that if you get close to a sick person, you're going to get sick yourself. Okay? But they never knew what was the particle and what's the agent that's transmitting the infection. And back in microscopy, I remember Steve was telling me when the first microscopes were developed. And so people knew these microorganisms, these little things like bacteria that were swimming around in pond water. But no one ever associated that with actually causing the disease. And so it was Pasteur who made the association that something about the disease must be transmitted by these little microorganisms that we're seeing in the microscope. Okay. And so he developed the germ theory. And so because of that discovery, now we know to sterilize food before we eat it. Okay, so either by treating it with heat, which is called pasteurization of dairy products, you're able now to kill the germs and prevent the spread of infectious disease. So two major concepts. As a result of vaccines and antibiotics, now diseases that used to be major killers in the past are history. Okay, they don't affect people anymore. So here in the United States, back in 1941, measles would kill almost 1 million people in the United States, whereas back in 1996, there were only 500 documented cases of measles. Right? Diphtheria and mumps, also similar stories, right, because of the success of vaccines. So you might ask yourself, why do we need to be concerned if we do have antibiotics and vaccines? And we have several reasons for needing to still be concerned about infectious disease. So one is that we always have new biological threats, and maybe you remember over the past couple of years, one disease called severe acute respiratory syndrome, or SARS, which happened in Asia, and then also anthrax, which was an agent used in bioterrorism. So these are pictures. This was the first microscope use of the coronavirus attacking cells. So the virus is actually these small particles that are surrounding the green cells. Okay. People in Asia were wearing masks because they just did not know what they were guarding themselves against. And this is a picture of anthrax spores, all right, in the filament form. And then this is a kid who's got cutaneous anthrax. So this is not anthrax inhaled in the skin, but inhaled in the lungs, but rather anthrax which is landed on your skin and colonized there. And then so new threats, and also the speed at which infectious disease can propagate these days makes it a modern day concern. So it used to be that people just didn't travel around so much. So when there was an outbreak of an infectious disease, it would be localized to whatever village or town that disease broke out in. But now, because of modern transportation, this actually is a picture or a graph taken from a Time magazine article that chronicled the outbreak of SARS. So basically how 249 people ultimately died around the world from SARS, all because they visited the same one hotel in Hong Kong. All right. It turned out that a researcher in China who was doing some basic work on early work on SARS had contracted the disease himself and then had gone to travel in Hong Kong, stayed at this hotel where all these other international travelers were staying, and all of them had gotten infected. And so they were able to trace this all back. And so due to modern day travel, basically, one infected person managed to caused the disease to spread to Ireland, Canada, US, Singapore, Bangkok, Vietnam. So it's pretty amazing, right? And so it's because of modern transportation that infectious diseases that may not have been a serious concern in the past become a serious concern now. More on the destructive power of germs. If you look at, what you would consider major human catastrophes, you would think of, for example, the atomic bomb in Hiroshima, which had killed something around 70,000 people. And then there was the Holocaust, which would have, by some records, was about 6 million fatalities. In 1918, there was this one version of the flu called the Spanish flu, which killed roughly more than 20 million people in 1 year. By the end of that epidemic outbreak, which took about 2 years to clear out, there was something more than 30 million people who had died. Roughly a third of the population of Europe had died from this one outbreak of infectious disease. And here is just a list of recent human epidemics, including the avian flu, SARS, anthrax, West Nile virus, Ebola, hemorrhagic fever, and meningococcal disease. They all have taken place within the last or the beginning of the century.
Can infects be passed between people by bleeding?
Yeah, it can. So the cutaneous anthrax that you saw, that one, if you don't wash your hands, yeah, you definitely can infect somebody. And pulmonary anthrax, whether or not somebody could get that, I'm not sure whether or not they need to be quarantined. It's possible.
Are the spores traveling through the air or the transfected?
Both can happen, yeah. Which is why the idea of sending anthrax in the mail could potentially work if they had done things right. Usually, when something is transmitted in the air, though, it's not really a free germ which is floating around, but rather it's inside a particle. because most of the time we're breathing out water vapor. So it's a microscopic droplet which contains several hundred germs, for example, inside of it, and that's what we breathe in. So it does still make sense to wear one of those masks, just a simple cloth mask on your face. Not that the pores are so small that it could trap an individual virus or bacteria, but rather if you assume that the germs are traveling on the water vapor particles, those particles may be about a micron in size, or several microns in diameter, so you should be able to put that out on the mask. I've given this talk to other groups, usually high school students, to explain what this is all about. And so just to give people the idea of what we're talking about, how infectious disease can spread so fast, I give them a simple math problem where supposing that we're in an auditorium with 100 people, say, and then one person in that room is sick, and then every day we encounter 10 people, say, right? Each one of us encounters 10 different people. So over the course of a week, how many people do you think could get exposed? And so I give them a multiple choice. Would it be, n the number of people in the auditorium times 10 people times 7 days, or would it be n times 10 to the 7th, or even more than that, right? And so most people are smart enough to realize it's not the first one. If anything, it's the second one, maybe even more, right? Because it's a geometric progression. And if you consider, like you said, that you could pass germs simply by coughing on your hands and then touching a doorknob, right? You don't have to be in direct contact with somebody, but if you leave a trail of germs behind you, then anybody else who enters that situation or environment is also going to get infected, so or at least get exposed. So really, of all the forms of bioterrorism, infectious disease is probably the most effective way to reach out and terrorize people. Infectious disease takes many forms. We talked about bacteria and viruses, but there's also fungi. And so examples of bacteria. So bacteria are the smallest single-celled organisms. So they are alive. Okay, they can grow outside of the human body. As long as they get food and nutrition and water, they can continue to divide by themselves. All right, examples of this include Staph aureus. This is actually responsible for acne. It's a bacteria which is on everybody's skin. Usually it's not common. E. coli, which is responsible for food poisonings. Bacillus anthrax. So these are all bacteria. Then viruses, bacteria in general are about a micron in diameter. Viruses are smaller than that, about 100 nanometers. And then these include things like influenza, variola, which causes smallpox, as well as HIV, that causes AIDS. Right? And these are packets of genetic material. So by themselves, they're not considered living organisms. If you had some in a test tube, they would not automatically multiply or divide. So it needs really to take over a host cell and make use of the host cell machinery to replicate. So by itself, the virus is not considered a living organism. Fungi, these are a little different, include mushroom, molds, and aspergillus. Not all of them are necessarily harmful to human health. But in some cases, when a person either gets a lot of exposure to fungi or their immune system is down, then this becomes an infectious disease. So I need to introduce you to immunology, right, before we get into the discussion of antibodies. And so the body does have some natural defenses against infectious disease. And there's three primary mechanisms in our body that are at work. One is our skin. Obviously, this is a physical barrier to the environment. And then we have also a cellular component as well as a molecular component. So we have white blood cells and we have antibodies. I think I have separate slides for this. So antibodies. These are special molecules inside the human body, which the specific role is to identify foreign materials that are inside. Generally, the immune system will attack any type of material which antibodies will attack. So you can think of antibodies as kind of a red flag. All right, antibodies are in your bloodstream, and any time it sees and recognizes something that doesn't normally belong inside the body, it'll stick to it. All right, and when antibodies stick to something, that is a trigger for the immune system to activate. All right, and do something about it. In healthy adults, you can carry 1 trillion of different antibodies. So these antibodies then are capable of recognizing many different objects inside the body. Right? And these are two different schematics of the antibody molecule.
So typically, an antibody is depicted as a Y-shaped molecule with two arms. And each one of these arms can recognize what's called an antigen. Right? An antigen is any kind of molecule that can generate an antibody response. And so these antigens all have unique shapes, right, and contours, which can fit onto the ends of each of these two Ys. And this is a ribbon diagram showing kind of the similarity between this drawing and the real molecule, what it would look like. And this is actually the IgG form of antibodies. So there are other forms of antibodies involved. Now, the other component of the immune system are the white blood cells. And these include macrophages, neutrophils, and lymphocytes. And each one of these play different roles in the body. And so there are a couple of ways that immune cells, for example, like macrophages, will work. One of them is to engulf any kind of foreign object. And so this is actually a video microscope, or this is a microscopic movie showing how a macrophage is crawling on the surface of a slide, and it smells these two yeast cells. And these yeast cells are called candida. And so it'll go and actually swallow up and eventually digest the yeast cells that are on the microscope side. Another strategy that immune cells work, and this is actually called a T. This is a T killer lymphocyte. Right? And what happens here is that this cell was infected with a virus, and so the T killer cell recognized that it was an infected cell, and to prevent that infected cell from producing more virus and spreading the disease, it decides to lyse it, or to basically pop it. And so this picture up here shows how a cytotoxic or T killer cell can actually kind of stab the host cell, the affected host cell, and cause that host cell to pop. Right? And once you cause that host cell to pop, it can no longer produce the viral particles. So it stops the infection disease. Any questions about that? Okay. So there is an ongoing war against infectious disease, at least in our body. The immune system is working constantly to keep infectious diseases in check. And so normally when our immune system is functioning, then the germs which are on our skin, in our bodies at this moment, right? We're healthy. We're relatively healthy right now, not because there's not a single germ on our body, but there is kind of a basic background load that our immune system can handle. And it's only when somebody coughs in our face, we get suddenly exposed to a tremendously larger amount of germ that we get infected, or that it's a cold day, for some reason our immune system is depressed, and again, kind of the balance tips in favor of the infectious disease taking over. Ultimately, then, in early detection of infectious disease is the key to avoiding exposure and to prevent a widespread epidemic outbreak. So now, with that brief introduction to infectious disease, we'll get into some basic methods of detection. Right. So the classic method of detecting germs is through laboratory culture and microscopy, but then there are two techniques that I'll talk over briefly. One of them is immunoassays, and the other one is DNA fingerprinting and hybridization. So laboratory culture and microscopy is the classic method. The basic idea is to take a sample of maybe sputum or blood or urine, whatever you think might contain the infectious germ, and then you put it in culture, right, in an incubator, and you see what grows out over time. And then once you have something that you believe has grown out, you look under the microscope and see what it is you can see. And so this is used to this day in modern hospitals. The advantage is that it's a gold standard. Right? All hospitals recognize this technique and use it. The disadvantage is that it's a long process. It takes 7 to 10 days. It requires a trained technician to interpret what they're seeing under the microscope, and not all of them have the same level of expertise. And often with some of today's more modern infectious diseases, a patient may not live 7 to 10 days for a diagnosis, and so you really need to treat that person before that.
nucleic acid probes, PCRs, and that more quicker to do. I mean, much quicker, right?
Yeah, and so that was the main revolution in infectious disease medicine, was the idea that instead of trying to detect the germ itself, you can detect just a particle or a piece of that germ. That would be enough, right? And so then enter the era of molecular detection, right? So that's what this is about.
Do they still do cultures, just kind of cheaper?
Yeah. Well, so that's the thing, is that before it was considered that molecular detection was this exotic technique that was only affordable by modern hospitals in the U.S. and would never be used in third world countries. But, stuff that we're working on at CBSC is trying to make biodetection cheaper and more affordable, right? So that, and that's what we'll get into with the class, is kind of techniques, what are some techniques to make this more efficient. But the idea that you can detect molecules, then that process will ultimately be faster and more accurate than trying to detect germs themselves. Was the main idea. And like you said, the advantage here is that it's extremely sensitive and you can get results much faster in the order of 1 to 3 hours instead of 7 to 10 days. All right? And a lot of these techniques, you don't even need a microscope to try and detect the germs. Basically, you're just looking for some trace or evidence that the germ is there without having to observe the germ directly. The disadvantage is that it's complex, it's not necessarily cheap, which means it's not universally affordable, and it may require some specialized training to use. The challenge of finding molecules in solution in biological samples is a lot like playing the game Where's Waldo. And I don't know if all of you are familiar with this, but it's basically the kid game is where you're given this picture and you're trying to identify this one particular individual. It's called Waldo. Okay, and so this is exactly analogous to trying to find a germ that might be inside blood or inside some kind of a lab sputum. All right, you're talking about a very complex solution where you have lots of proteins, possibly DNA, possibly other materials, and you're trying to pick out that one molecule that belongs to anthrax or belongs to E. coli. I don't know what we're going to do that because many things look alike. All right, and here, I think this was taken off the web, and I think the hitch here was that Waldo didn't really exist. There was really no Waldo here. I plugged it in. I think I put Waldo here.
No, that's not Waldo. That's I think it's not Waldo because I was looking at this picture and I couldn't find him. I had to look everywhere.
So anyway,
you think it's Waldo?
Yeah.
Because he looks a little bit weird there. He
So
looks like he has like a mustache.
Right.
Like I think he also has like a brother, Outlaw, which
Really?
is Waldo read out backwards.
Oh, I see. Outlaw. Okay. So you might ask yourself, what could you do to make this faster or easier? All right. And one trick would be to paint Waldo purple. Okay, because even in this kind of mix, if you painted one guy purple, at least it'd be easier to spot. And this is analogous to doing conventional staining of tissue samples. Okay, when you saw previously in Steve's presentation where you're looking at tissue which has been stained, all right, that's a visible stain, meaning that in transmission microscopy, if you look at an unstained cell, really there wouldn't be a whole lot of features. You might see an outline of the cell, maybe an outline of the nucleus, but that's about it. Okay, if you wanted to see something more, you wanted to see mitochondria, you wanted to see lysosomes, you wanted to see the cytoskeleton, you'd actually have to stain it so that it was a different color. Okay, and so that's what you'd be doing here, is that you'd be staining Waldo with a particular color. Now, it kind of helps, right? I mean, if you kind of look carefully, it's like there's only one purple person there, so you spotted it. You kind of made it easier. But it's still not, I mean, you still need to hunt around. All right. The problem is that you're not achieving very good contrast. And so the other strategy is to make it glow in the dark. Okay. Because if you make something glow in the dark, same way if the lights were on and I had painted a green dot on the wall, you may not be able to spot it. But in a darkened room, if I were to shine my laser pointer on the wall, you'd be able to spot that bright green spot, no problem. It's because of the contrast between the laser spot and the background. All right. In the case of visible microscopy, right, you might have a distinctive color, all right, but you'd have to do wavelength discrimination because the background basically is very high relative to your signal. Whereas this way, the background is very low relative to the signal. And so this is the whole rationale behind doing fluorescent tags as opposed to doing visible dye stain. Okay. But, how do you implement this? This is all great in theory, but how do you implement this in practice? Because what you're doing here is you're taking Waldo, you're painting him, and then you're throwing him back in the crowd and saying, oh, I see him, he's right there. All right? Which is kind of defeats the purpose, because when you're doing, say, cell imaging and microscopy, you can't take the object that you're looking for, pick it up, paint it, and then throw it back in there to say, oh, there it is. Because if you've already picked it up, then you've accomplished your goal. All right? There's no point in detecting something if you already have it. And so how do you attach a fluorescent dye to something that you don't know what, even if it's there, right? And so for this, basically you have to go back to immunology and make use of the antibodies that we were talking about. Okay? And so this is now the introduction to immunoassays. So the idea is how do you make a special paint for a dye that will magically fly to your object in question, to your target. And the idea is to purify, is to take antibodies, right, that will specifically bind to your target molecule. You want to be able to purify that and then conjugate that to a fluorescent molecule, so that bifunctional molecule now is able to not only bind to whatever target molecule you're looking for, but also it'll be carrying a flag that you can see from a distance.
How do you isolate it from everything else in the board?
Yeah, so this would have been something that I would have liked to have a separate slide for. But basically, you'd be purifying the antibody from whatever organism had antibodies against it. Okay. And so in the laboratory, typically what you'll do is you'll inoculate. So for example, let's say pick some antibody that you want to develop. You want an antibody against anthrax? Sure. Okay, so suppose you wanted an antibody against anthrax. Most likely you don't want to find some human volunteer to inject anthrax into, so you'd find either a chicken or a bunny rabbit. Now this is to develop an in vitro test, or a laboratory test. Okay. You would take a chicken, for example, and inject anthrax into it. Now the chicken will respond, it'll get sick with anthrax, but it's also, the chicken will develop antibodies against anthrax. And then you take a sample of that chicken's blood and you can purify the antibodies, and most of the antibodies in there will be directed against anthrax. And so that's a normal response.
Well, how do you purify just antibodies based on everything else that's in that?
So you would use affinity chromatography, for example, and so you could have an affinity column which had anthrax, ground up anthrax attached to the column. Okay,
I see.
And then I pour the chicken blood over it. And now the antibodies that are inside that blood will stick to the anthrax. Okay. And then I do a wash, and now everything that's not sticking to the anthrax will be washed away. All right. And then you can elute the columns with either acid or some kind of organic solvent, and then that will cause the anti-anthrax antibodies to release from the anthrax and be collected in a cup. And now I have a cup that contains a solution with just anti-anthrax antibodies. So that's the basic technique. All right, and what you're doing there is you're collecting polyclonal antibodies against anthrax. Okay. So that's like the most crude form of obtaining antibodies. So if you were in a desperate bind and you needed to get that time, you could. Does that make sense? So assume now that you have a container, a test tube containing these antibodies. There are chemistries that allow you to couple the antibody to the fluorescent tag. Right? Now, you may have heard of things like fluorescent isopathinate, CUTC, or tetrarotamine isopathinate, CUTC. Right? So the ITC of CUTC and TRITC, that's the chemical group that actually allows you to covalently couple the fluorescent molecule to any free amino groups that are on the antibody. Okay, so there's chemicals for doing that, but then basically what you end up with at the end of the derivation is a complex molecule that's half antibody, half fluorescent molecule, and those things, since it's covalently coupled, is fairly stable, and it has all the binding affinity of antibodies, and it has a fluorescent label. So it's something you have to keep in the refrigerator to prevent the antibody from breaking down. You also have to keep it away from light to prevent the fluorescent part from photo-bleaching. But you have that reagent, and now you can use the reagents in a number of different tests. And so the test here that we're showing you is the plastic sandwich assay. And this is basically a method to determine yes or no, is there some kind of target molecule present in my biological sample. And I'll just move forward to this assay. So the test would take place originally before introducing the fluorescence component. The original method of using the antibodies is called enzyme-linked immunofluorescence assay. And so the idea here is to create a special laboratory culture dish, all right, that have these individual wells. In the bottom of each well, you would have these antibodies that are stuck to the bottom. Okay, and these antibodies are directed against whatever it is you're interested in. In this case, this could be something that was going to recognize HIV. All right, and so these are anti-HIV antibodies that are attached to the bottom of the culture dish. Now, if we were concerned if your bottom was exposed or if you were exposed to HIV and did you already have this virus, we could take a sample of your blood and put the sample of blood inside each one of these wells. Now, if you have HIV in those samples, then the HIV would stick to the bottom of the well, like you see here. All right? And then you'd have to do a wash to get rid of all the other stuff that's in your blood. And now you go in again, okay, this time with HIV antibodies that contain the fluorescent tag, but in this case it's a special enzyme that can induce a color change. Okay. And again, only if the HIV particles are present inside that sample, you'll be able to couple the antibodies on the bottom with the antibodies on top. And now if you add the final reagent, which changes color in response to this enzyme that's attached here, you'll see this kind of effect. All right, and with basically, we start out with a solution that's colorless, but then if the enzyme here is present, then that solution will go from clear to green. All right, and the darker the green, the worse the news, because it means you have a lot of HIV present. Okay, so this is kind of the classic laboratory technique, and this basically allows you to say yes or no is HIV present in the sample without needing a microscope. You don't need a super resolution microscope to try and see the HIV virus itself. Okay, what you're looking for is the presence or absence of the HIV molecule, a molecule associated with HIV, using this kind of scheme. The whole process can be done in roughly 2 hours, 2-3 hours. And so it gives you results much faster than if you were to do laboratory culture.
How accurate could you actually detect something?
Yeah,
what concentration?
So we'll get into this, but basically the accuracy depends on the antibody, right, ultimately. So if you have a good antibody, then you'll have a good test. But if you have a bad antibody, then the test is bad. So yeah, we'll get into a little more into that. Any questions on how that works? So with immunofluorescence, what we're doing is we're swapping out this tiny component. All right. So this is an enzyme that causes it's horseradish peroxidase that causes this dye molecule to go from clear to trim, to go from clear to opaque. All right. Once the peroxidase acts on it.
Is it attached to the?
Yeah.
Just horseradish.
No. So horseradish peroxidase is this object here.
Uh-huh.
Okay. And the horseradish peroxidase is attached to this antibody. So often we use two conventions. So we call these the capture antibodies, and we call these the detection antibodies. Mm-hmm. So the capture antibodies stay attached to the bottom of the plate. The detector antibodies tell you yes or no, did the capture antibody successfully catch them? Okay.
I think the thing that causes the fluorescence to start is like a chemical.
So if this were fluorescence, then what you would be looking for were any of these wells that were fluorescent. Okay. So fluorescence will occur only if that target, whatever it is you're looking for, is present. Because if your target isn't present, there's no reason for these antibodies to remain attached to the bottom of the well.
But if one target binding, they undergo the conformational change.
You're talking about something which is a little more advanced. So that would be something like a molecular beacon. Right? So the idea is that something doesn't produce a signal until it's successfully attached. Yeah. Okay. The problem is that with the old-fashioned molecular probes, the old-fashioned fluorescent antibodies, they're fluorescent all the time.
And they only stick.
but they only stick if the target is present. So this is why it's so important to do a wash. Because if you don't wash, then all of the wells are going to have the fluorescent antibodies, and they're going to say, oh my God, it's all positive. But if you didn't do a good wash, then you have stuff that's sticking there non-specifically. All right. And so that's the reason why these techniques are not the most advanced or not the most ideal. Okay, it's an improvement, but it's still not perfect. Okay, and that's because it requires that you do washes in between these different steps. So you start out with just the empty wells. You add your sample, and if the target is present, it will stick to the bottom. But if there's nothing there, then nothing will stick. So after you do your wash, you end up with this again. Okay, and if you end up with this, and I add my detection antibodies, well, they're not going to stick to the bottom either because there's nothing for them to stick to. So that's why this is called a sandwich assay, because there are different layers. Okay, 2 pieces of bread with the meat stuck in the middle. And so in this case, the 2 bread layers are the layers that contain the antibodies, and the meat in the middle basically is your target, or your HIV or your anthrax, whatever it is you're looking for. Okay. And it's only the 2 pieces of bread don't stick to each other unless there's a middle. Okay. If there's nothing in the middle, then when you do your wash, the top layer just washes off. Okay. So you only see fluorescence and you only see a color change when the target is present. If the target's not present, you don't see fluorescence, you don't see a color change.
So that's the only limitation, though, is just having to do a wash. It's still pretty good. I mean, that's not a huge,
we'll talk about that.
Just a step.
So again, like we said before, it's faster than doing laboratory culture and microscopy, and it allows you to get a positive test without having to directly observe the bacteria or the virus. Right? The disadvantages, well, you might ask whether or not it's easy to use and whether it's foolproof, and then the accuracy, how do you know or prove that the test is actually correct? And so this is actually the home pregnancy test is an example of immunofluorescence.
What antibody do you use in that?
So that one, they're using an antibody against what's called human beta HCG, human chorionic gonadotropin. So this is actually a female hormone that goes up when a woman is pregnant. Right? The hormone itself is a protein, and it starts accumulating, and it shows up in the woman's urine. So the woman pees on the stick, and if there's any beta HCG that's present in the urine, it'll stick to the antibodies on that stick.
So that's using ELISA too.
Yeah, essentially ELISA. And so then the stripes that will appear, plus or minus, basically it's that third component, the detector antibody, right? And there it's once you've activated it, you've gotten it wet, so it releases the detection antibody and it just flows over the surface, right? And so then you get a color change. So that's essentially how that works. So yeah, to get into the question about the quality of the test, like Elon was asking, we do this type of analysis. Have you talked about truth tables, or how to analyze different laboratory tests, sensitivity and specificity? Okay, so in case you haven't, there are many ways to evaluate a laboratory test in terms of whether it's a good test or a bad test. The way that we often will describe a test is in terms of the test sensitivity or its specificity. And so the way that you can break it down is that you might have the test result come out to be positive or negative, and then in reality it's positive or negative. So you could end up with 4 different outcomes. In one case, you have a disease, you really do have a disease, okay, and the test says yes, you have a disease. In that case, that's a true positive. Okay. It could be that you really don't have a disease, and true enough, the test tells you, nope, you don't have a disease. So that would be a true negative. But the test could go wrong. It might say that you have a test. It says the test is positive, but you don't have a disease. That would be a false positive. Or the test says you don't have the disease, but you actually do have a disease. That would be a false negative. Does that make sense? I know every time I talk about this, I mess up what I'm saying. So what I mean is that you can have a true positive, a true negative, a false positive, a false negative. Okay. And then the sensitivity of the test is basically how often the test is correct in saying that you're positive. No, that's not right. How many times does it pick up? Right, out of all people who truly have a disease, how many people does it pick up? Okay, that's sensitivity. So something which is very sensitive will pick up the disease in every single person who has the disease. Okay, that's something which is 100% sensitive. Specific means how many people will it say, how accurate is it when it tells you that you don't have a disease? Right? If people don't have a disease and it says it's negative, then it's specific. Right? So you can think about this, all right, to see whether or not you believe all of this. But basically, these two are not redundant. Okay, you need to know both. Okay, so because I can have a test which is just basically stuck in the on position all the time. Okay, so no matter what, it's always telling me positive. That's 100% specific, but it's 0% sensitive. I'm sorry, it's 100% sensitive, but 0% specific. See, I knew I would say that. Right? As opposed to a test which is basically always negative. All right? Something which is always negative would be 100% specific, but 0% sensitive. Right? So there's value in knowing the two. Okay.
Is this the same thing as a type 1 and type 2 error?
Yeah. So I think this is the type 1 error, and this is the type 2 error. Right. But that we need to go into that statistics to talk about that.
I mean, just basically the bias. What was the bias theory? The bias. Yeah.
Bias theory.
Bias. Yeah.
No. This is not. This would not be bias. So this is simply.
If you test so many and so many people with the test which has such a
So there's certain situations where the sensitivity or specificity will go up or down depending on the prevalence of a disease. So if you're sampling in a population where everybody is infected, okay, then it's impossible to get good specificity because you have no one in your sample is non-infected. Everybody is infected. So it's very difficult to assess the value of the test sensitivity or specificity in strange environments like that. Or also in the case where the disease is extremely rare, 1 out of 1 million people have it. And so then you can't test the sensitivity of the test. So limitations of an antibody-based test is that the test is only as good as the antibodies that you use. Certain antibodies can recognize more than one antigen. And again, I go back to the original drawing, realize that these antibodies are only looking at the shape, and so more than one shape can sometimes fit in an antibody. So an antibody might be most specific for one particular shape, but doesn't mean that it won't accept or bind to other shapes that are similar. And so antibodies are never 100% specific. They're just one type of molecule. The ability of an antibody to bind to antigen depends on the concentration of both the antigen and the antibody that are in your sample. There's a whole area of molecular binding that you get into when you're talking about biophysics. And so I'm not going to get into that. But basically realize that whether or not an antibody binds depends on the concentration of the target, depends on the concentration of the antibody. And so there's something called a binding constant. It's also called an dissociation constant, which is usually the concentration of antigen that's required in order to achieve 50% binding of the antibody to its target. And so when you're describing the quality of an antibody, oftentimes a company will describe its antibody in terms of the binding constant. And a good antibody, or what they consider to be a good antibody, is one that has a very low binding constant, meaning that at very low concentrations to the target, your antibody is already 50% bound. Okay, something which is weakly binding, you need a lot of target, you need a lot of antigen present in order for that antibody to achieve 50% binding. Okay, so sometimes that doesn't make sense to everybody. So if you have any questions, does it make sense to you? Okay. So usually in commercially available antibodies, you can get at least micromolar, if not nanomolar sensitivity. And you might think nanomolar sensitivity is very good, but if you think about what we're doing with molecular detection and doing single molecule studies, you have to remember that molar unit is based on Avogadro's number, right, which is 6.023 times 10 to the 23rd molecules per mole. And so even a nanomole of sample, right, still corresponds to a huge number of molecules. roughly 600 billion molecules in a nanomole of solution. So, in terms of if you still need a lot of target in your sample in order for antibodies to bind. So, what can we do to improve on immunofluorescence or immunodetection? One is to use better antibodies, and so I didn't get into monoclonals or synthetic antibodies, but there are new strategies to create these really like super strong antibodies that'll recognize even like a single molecule of target. And there are methods to do that. You can also consider other molecule binding pairs. So what I've talked about so far is just the antibody-antigen relationship. Right? So this is one where we're making use of immunology to find two pairs of molecules that bind to each other, or one pair of molecules that bind to each other, an antibody and antigen. But then there are other methods as well, and I'll talk a little bit about it, genetic mechanisms, where you use the two strands of DNA, right? So this would be genetic detection, where now you use the sense versus the anti-stress. If you're looking for one, you use the common in order to find a natural molecule that will search for and bind to your target. You can also make use of the enzyme-substrate relationship. So this is an enzyme that naturally looks for some protein or target that it would recognize and bind to it, right? And normally it would chew it up or cause something to happen. But you're just interested in that first component where the enzyme will search for the substrate, or the substrate will search for the enzyme, right? And you can couple a fluorescent molecule to one or the other in order to search for the complement. And then also a receptor ligand, so you could find some kind of a receptor for a particular hormone and use that receptor as a hook, basically to fish around for the hormone. Or you might do it the other way around. So you might have a hormone and you're looking for the receptor. So that you attach the fluorescent molecule to your hormone and then look for the receptor in the cell and do it that. So these are all different techniques to try and develop a better approach for molecular detection. And then another trick is to simply amplify your signal. All right, you can do it either before or after binding. So if you wanted to do it before binding, you would do something to try and amplify your sample, and that's what DNA PCR is all about. Right? And so in the case of genetic detection, what you might be looking for is yes or no, is a particular gene being expressed, or if you have a particular gene that is coupled to a disease. And so in that case, the gene in question, you would basically do this process of, I don't have the PCR. Well, so this is a PCR machine, but I don't have actually the diagram for PCR. But PCR is basically polymerase chain reaction. The idea is that you use this special enzyme that causes DNA to replicate itself. And this enzyme functions at high temperature. And you can use this device that basically controls the temperature and causes the DNA that's present in your sample to amplify itself, so that now, instead of having to detect just one copy in your sample, you can detect hundreds or thousands of copies of that sample, of that particular gene in your sample. And then you use as your molecular probe a strand of the antisense DNA or RNA to look for the presence or absence of that sample that hopefully you've amplified. And so that would be the basic concept of DNA detection. And that's been now carried out here in terms of microarray technologies and gene chips, where these are silicon-based wafers that you can functionalize with a strand of DNA. So it could be that you're searching for a particular sequence of DNA, and so each of these elements on the array is attached with a different sequence of DNA. Okay, and now if you were to pour a sample of biological liquid over it, and you wanted to say yes or no, is this sequence present, is this sequence present, is this sequence present, then you simply look for hybridization to occur at any of these test elements. And if you got successful binding, now you might see fluorescence on that spot, or you might see a color change on that spot. So it's kind of similar to ELISA, except instead of relying on the antibody-antigen binding relationship, now you're relying on DNA hybridization to allow for your capture and detection molecules to couple to each other.
But how do you read then what happens? So if you have this hybridization, where comes the fluorescence?
Oh yeah, So yeah, that's a good point. So here it's usually there's usually a scheme where before you add your unknown mixture, you basically added a fluorescent tag to all the nucleic acids in that sample. So now that nucleic acid is tagged. Okay. So regardless of whether it's the sequence that you're looking for, if any of the DNA that was in your sample attaches to the DNA which was on your gene chip, now you see fluorescence over that element. And again, washing is an essential. If you don't wash, you may get all these false positives in your sample. Okay. I just wanted to, we're almost 10 minutes away, so I just wanted to explain that we have been developing new technologies to improve on molecular detection, particularly with immunofluorescence. And this was using a system that was built by Luminex Technologies in Texas. And the idea here is to take the ELISA assay and to put it on the surface of a microbead. Right. And so here, instead of the bottom of a well, now we're talking about the surface of the microbead. The microbead is about 10 microns in diameter. And so these would be the capture antibodies. If you were to mix this bead in with a biological sample that might have anthrax or HIV, then now the virus would stick to the surface of the bead, and now you add the capture antibodies. And the capture antibodies are tagged with some kind of fluorescent molecule so that the bead lights up only if the target is attached to the outside. If there's no target, then these beads are not fluorescent. So that would be the idea. And then you could analyze this in a flow cytometer. The other thing I forgot to mention is that each of these beads can be color-coded, so that each bead is responsible for recognizing one type of germ or another. Okay, so an orange bead might be for anthrax, or a red bead might be for influenza. Okay, so every color bead represents a different test for a different kind of germ or infectious disease. And so all of this can be analyzed in a flow cytometer. And in a flow cytometer, basically, you would be interrogating two components of the bead. So one is yes or no, did that particular bead, whatever it is specific for, capture something? You would look for fluorescence on the outside, so then you would use one color laser to see yes or no, is there fluorescence. And the other one would be to say, well, okay, if there is fluorescence on the outside, tell me what was that bead originally supposed to capture? Was it supposed to capture HIV? Was it supposed to capture anthrax? What is it? Okay, so this one tells you which bead it is, and this one tells you whether or not it caught something. Okay, and then the flow cytometer, and these are actually on a microscope slide. I put these beads down, and then you can see that in fluorescence, you can not all of the beads light up. Here, I needed these two colors in order to tell me what bead class it was, but this told me whether or not it was fluorescent. Okay, but basically, with these pictures, I could read out and tell you in a particular complex sample what germs is containing. And so ultimately, we had created this system over at Livermore, which was the autonomous pathogen detection system. The original device was here. This is a flow cytometer that it basically will analyze all the individual beads through a capillary system. So in a capillary, all the beads would march through a single file, and these lasers would hit the beads, and basically you'd gather up all these statistics on about 10,000 microbeads to give you basically a profile of what are all the different molecules this particular biological sample contained. So we took that machine and then built it into this device here, which would continually sample the air. So this was essentially a smoke alarm, but not to detect smoke, but to detect any kind of aerosolized bacteria or pathogen that was in the air. You would take that air sample, bubble it through water, and then you would sample that water to see whether or not there were any germs present inside of it. So that was the end of the presentation that I had originally made. And then in terms of other molecular probes, which you'll be hearing about in later talks, instead of using fluorescence. So I talked here about what are the different biological tools that we can use to find the molecules. Now we can focus more on what are the different optical properties or techniques that we can utilize to create better labels. All right. And so we talked about fluorescence and we talked about visible dyes, but there are other techniques as well. So one of them is bioluminescence. So bioluminescence, as opposed to fluorescence, is a different way of producing light. So fluorescence is produced, it's basically light energy being converted to more light energy. So you use light at one wavelength, usually ultraviolet, to create light which is visible wavelength. Right? But bioluminescence or chemiluminescence is more appropriate, is light which is created through a chemical reaction. And in this case, this is what's, you'll hear about this because we developed a test for prostate-specific antigen, PSA, with this. But bioluminescence is what fireflies use to create light. And so this is a reaction between an enzyme and a substrate, namely luciferin and luciferase. Luciferase is the enzyme, luciferin is the substrate. And in this case, bioluminescence is great because most mammalian systems don't have anything which is bioluminescent, as opposed to fluorescence, where we have a lot of molecules in our body which are autofluorescent, meaning that if we were to ever develop a test using fluorescence, we have to contend with a high background, because there are a lot of other molecules which are fluorescent. So we're not sure whether or not our fluorescent signal is specific, because we might be seeing fluorescence that might be coming from something else.
So we have a lot of fluorescent potential, we just don't have any fluorescent enzymes to catalyze the reaction.
Right. But you're right. And so so with bioluminescence, we don't have any bioluminescent enzymes.
Yeah, but we have substrates that would, if we had the enzymes, that would actually
sort of. We have neither, basically. So since we don't have the enzyme or the substrate, we just don't deal with it.
I thought that's what you said. Okay.
No. So the idea here is that we can couple basically, and it's probably better for us to save this for the next talk, the later talk. There's going to be a specific talk about this. But the idea here is that we can modify the substrate so that you can add a peptide to the luciferin substrate to block the light reaction. Okay, and when you block the light reaction, now you're testing to see whether or not, for example, serum protease in the bloodstream is present or not. That serum protease will feed that additional peptide off, and now suddenly the light reaction can proceed. So basically, this is a way for us to use bioluminescence to detect yes or no if you have some particular serum protease active in your blood.
But would it be toxic if you?
So this is not an in vivo assay; it's an in vitro assay. So something outside of the body. All I do is take a sample of your blood and perform a test on the lab bench. So it's not something that I'm going to take this engineered protein and inject it into the transfusion. Yeah, that would be bad. So that's one area of molecular probes that I didn't get a chance to talk about. What Melissa was talking about molecular beacons is also a very big field, which we can get into. But the idea here is how do you create a molecular tag which basically doesn't stay on all the time? Okay, because what you've seen with fluorescent antibodies, basically, even before I've used it in my test, if I were to just shine ultraviolet light on a sample with my fluorescent tag, it's going to fluoresce. Okay, so it's on all the time. And there are times where I can't do a watch. For example, I'm trying to detect a tumor that's in your body, and I would like to use a fluorescent label to inject into you and have that fluorescent label go to the cancer. Well, unless I have a way to wash out the non-specifically bound label, I won't be able to detect where your tumor is because there's fluorescent stain everywhere else in your body. Right?
Well, maybe you can.
Well, maybe you can, right? Maybe you can, but then this would require somebody who knows enough histology to say, well, that's not supposed to be there. That doesn't make sense. So that's obviously non-specific or irrelevant. Whereas this looks definitely like this is a tumor. Right? And so being able to distinguish signal from noise is a big problem. Molecular beacons is a whole area of research, and the idea here is to do something where a conformational change causes your probe to go from dark to light. Okay, so something which is dark until it binds to its target, and then suddenly lights up. And so here, one strategy is to use what's called fluorescence quenching. Right? And the idea here is that a fluorescence molecule will fluoresce as long as there's not another molecule nearby that could take away the energy that normally it would use to create a photon. Okay, and so I could take fluorescein, for example, and bring to it a metallic nanoparticle, a gold nanoparticle. So if I bring gold close to fluorescein, that fluorescein won't fluoresce because the gold is absorbing all the energy that normally that fluorescein would use to create light. Okay. Now, if those two objects are close together initially, but now so I create a synthetic molecule that basically allows those two objects to be close to each other. Now, when that molecule binds to whatever it is it's looking for, suddenly it causes the two parts to dissociate. So now suddenly the fluorescein is away from the iron or the gold, and now fluorescein is free to fluoresce. Now, this is great because it doesn't fluoresce unless it successfully found its target. And now I don't need to watch, right? Because anything which did not successfully bind to its target still remains in the closed position. And so it could be somewhere else in the cell or somewhere else in the body, and I don't need to worry about accidentally seeing its fluorescence, because there's no way it would fluoresce unless it had successfully bound to its target. So that's kind of the holy grail of molecular probes, is to create something that can do stuff like that. And with DNA, it's the same idea, is that you can go in with actually a pair. So instead of using just single-strand DNA or RNA, you use double-strand. Okay, but you use a double-strand where now your target will successfully compete away the opposing strand which you injected. Okay, and so you originally inject something which is double-stranded, where you have a fluorescent molecule on one strand and you have a sequencer on the other strand. Right, and then when that thing goes in and successfully finds whatever gene it is in question and successfully competes away the strand that contains the quencher, now suddenly the strand that contains the fluorescent molecule is free to fluoresce. So again, you've got a beacon that fluoresces only when it's successfully located its target. Right? So there are other techniques as well, but that's generally the idea. What else was it? I didn't get a chance to talk about quantum dots, but that's another source of fluorescent molecules that you can use instead of using organic fluorescent molecules. And then I also didn't talk about GFP, and so fluorescent proteins. This is another big area of fluorescence labeling, which is totally different. And so as opposed to using fluorescent antibodies or fluorescent DNA or any other, so these are all labels that you need to inject into your sample or add from the outside. Okay, GFP represents a class of what we call fluorescent proteins. So these are fluorescent on synthesis. So basically here, instead of having to worry about how do I get my label to the place where a particular protein in question is located, now I modify the protein at the genetic level so that it's naturally fluorescent. Right? And so I don't need to worry about labeling it. Basically, as soon as that cell or organism starts to grow up, it's automatically creating that particular target that's fluorescent. Right? Now, I may not be able to use this to diagnose diseases in humans because you can't modify a human to create those kind of fluorescent proteins. But at least in laboratory animals, you can do this. And so, for example, we're doing the HIV studies this way by modifying the HIV so that it naturally produces a fluorescent molecule. And I don't have to go in with a fluorescent antibody to tag it. The other problem too is I didn't get into talking about sizes of molecules. So typically a fluorescent antibody is very large, and if you're doing studies like quenching or fluorescence resonance energy transfer, the size of the antibody may be so big that in itself is enough to block any kind of fluorescence resonance energy transfer. So if you're trying to do proximity studies, trying to show how two different fluorescent molecules might be co-localized, the presence or absence of an antibody could make the difference in making that determination. So it's a big field, right? But hopefully at least it gives you some sense of how much stuff is going on and how much thinking is involved with doing fluorescent tagging. All right. Any questions? Okay. So Thursday we'll resume with the normal schedule. I think Jack Rutledge is going to talk. So Colin, you may be excused this time.
No, it's all right. I'll be here. I like listening.
Okay. All right. So, good. Thanks.